Everything's working. Breathing away.
Thank you so much.
It's also recording locally too.
I'm really sweaty, don't hug me.
I'll risk it.
Okay.
Oh, wow.
Thank you. Thank you. Five minutes. Okay.
Yeah. It's okay for us.
You should.
I left my banana leaf at home.
If you do that, during the whole lecture, I might go and get one.
One on either side.
Somebody have grapes.
That's fine.
Right? For me.
Grapes?
Yeah, he said if you brought his banana leaf, he could ban me during the presentation.
That's a little too relaxed. You'll be relaxed.
If it's lying down.
The day bed, did we get the day bed?
The day bed?
Day bed?
What's the day bed?
Lounge.
Oh, like that kind of day bed.
How are you doing? It's all coming together. It's all coming together.
I paid them all.
Yeah, I didn't know if that was
Not a lot. I'm gonna do one more bathroom break. So I don't have to go in this.
Did you get your bag?
What? Thank you. You got my bag?
Yes, that's nice. But don't try it. Yes. That's right.
one of the
I'm talking about
You made it.
I'm going to be here for 15 minutes and then leave.
wait a year.
We might squeeze it out this
We're still waiting for Tom. Sorry? Tom Odell? Oh, no, he's there.
This is really
What's that?
This is really tight.
Yeah. So I don't know how to turn it down.
No, it's projected.
Oh.
This is just like.
Oh, yeah, sorry.
Okay, since it's 3 o'clock, let's start today an exciting event. First of all, I would like to thank you all for coming, family, a lot of friends, people from the program, for the NSIPT program. So welcome all. For those of you who come from the outside, this is one of the first tests we give to the graduate students to say, show up in CHS in that place, and they'll have to find it. So you all passed, those of you who are here, so you can apply for the PhD program. If you didn't get here, then then you didn't pass. So, anyway, so it's a real pleasure to introduce, and I guess many of you know Kathy already, so I don't really have to introduce her. But Kathy got her bachelor's degree in the University of Maryland in microbiology. And then she sort of felt that maybe microbiology at that time wasn't kind of the hot field that she really was interested in. And went to work at Columbia and first worked with somebody that probably all of you know called James Rothman, won the Nobel Prize a couple of years ago, and then decided neuroscience was really something exciting, and did an additional master's degree at Columbia, at that time working with Yamamoto, and really got introduced into diseases of the nervous system, neurodegeneration, and things like that. She came to UCLA, first went through the Access program, and then joined my lab, and also joined the neuroscience IDP. And she's really been a wonderful person in the lab. She's been in the lab for a long time. I think when I compare her with previous graduate students, she was the most independent graduate student, I would say. She really shaped her own work. She came in, she sort of took over, did things that had never been done in my lab before. She was the first one to actually amplify a plasmid in the lab, which we hadn't done before. She got us into the 21st century, right? And did it all by herself because I wouldn't know how to do that, right? And so it was really wonderful to have her. Has a great sense of humor, as you all know, and so that's why I left out any of the usual humor slides, because she is humorous, and for those of you who sort of want to hear her and live her own humor at the end, there's going to be a reception, not in here, but a reception that's mainly organized by friends and for her fiancé, who sits right down here. So it's down in the botanical garden. Those of you who don't know where that is, just hook on to one of us, and it's just basically down these steps over the street, and then walk a little bit into the garden, just basically in this direction, and here for the champagne bottles to pop, and we will be in the right place. So before we start, I also just want to give you just a brief way in which this works. So Kathy is going to give her talk. Then you guys can ask whatever questions you want. That is the public part of the defense, and then we'll ask you all to leave and take your way down to the botanical garden, except for the thesis committee. I forgot to thank you all for coming, because otherwise we couldn't start. So thank you for all being here. So the thesis committee is then going to ask Kathy some more questions. And the way that works for you is that we're first going to ask you to leave and talk about ourselves, all the good and bad parts. We usually just start talking about our last vacation or that really cool experiment. Then we ask you to come back in and really ask questions. And we ask you to leave again, and then we ask you to come back in. So don't be surprised. That's just the way it goes. And so without further ado, thank you very much.
Thank you so much for that kind introduction. And just checking that everybody can hear me. This is working? Okay, perfect. So I'm Kathy. I think hopefully everybody knows who I am. I am a graduate student in Dr. Felix Schweitzer's lab here at UCLA. And as you mentioned, they started here in fall 2010 in the Access program. So I have some of my Access colleagues here, and then after joining his lab in June of 2011, joined the neuroscience program, and now have some of my awesome neuroscience friends here, and other people that very generously offered to stop by who've been part of my graduate experience, and I really appreciate that you all are here. Thank you. And I'm a little clumsy, so move that out of the way. Not important on my laptop. Okay, so today I'm going to be talking to you about the work that I've been doing for the last 5 years. And this is really focused on understanding how presynaptic function works and how the modulation of the release at presynaptic sites works. And so then the subtitle of my talk is Rural Protein Ubiquitination and Effects of Environmental Toxins. So I'll talk about presynaptic function in the context of those two things. So the talk will be organized by the introduction, and then I'm going to talk for a while about ubiquitination, pesticides, and then briefly touch on some conclusions and future directions. So the synapse is really the major junction of information transfer in the nervous system. And I hesitated actually at first in putting this picture in because I always put this in my talks. I really love this image. This is the frog neuromuscular junction, and this is an electron microscopy image that was taken in, well, it was published in 1981. I don't know exactly when it was taken, but it's the first image that actually really proved that synaptic vesicles were the units of that were containing and transferring neurotransmitter at synapses. So essentially, this is the presynaptic site here where the synaptic vesicles are, and then this is the synaptic cleft and the postsynaptic region. And so this is the site at which information is being transferred from one neuron into another neuron. And so this is what I'm really interested in understanding. So how are synapses modulated? So in my cartoon here, this is that presynaptic site that I showed you, and this is also referred to as the axonal duton, and these are the synaptic vesicles full of little blue dots or neurotransmitter. And what happens is essentially this neurotransmitter is directed into the synaptic cleft when synaptic vesicles fuse with the plasma membrane. Then the postsynaptic receptors detect that neurotransmitter, and there's downstream signaling or processing that occurs. So this is the postsynaptic or dendritic spine. So it's just sort of a way of the context of thinking about where changes occur. In the presynaptic site, we would think about how vesicles get released and how they get recycled. So you see not only are there little blue dots coming out of this vesicle, but then there's this also guy that starts budding out here, and there's a recycling at this local site. And then in the postsynaptic compartment, you think about the number of receptors that are going to detect neurotransmitter and the actual properties of conductance of these guys. So we know there's a number of different ways that these sites are regulated. Messenger RNA trafficking, which is important for protein synthesis and protein turnover. So all the proteins, both of these sites, have to be regularly degraded and either transferred back into the region or locally synthesized. There are changes in the excitability due to ion channel expression, and this can change in response to environmental needs. So you would have different ion channels at higher expression, as well as modifications or the subunits of those channels that would change the properties of excitability. And then finally, we have post-translational modifications, which means that you have your protein that's already in one of these sites, and you add something to it that modifies its activity. So that could be a phosphate group, which is the protein phosphorylation point here, or you can add a small protein called ubiquitin, and this is what I'm going to talk about here. So, and this is, I forgot to mention, and this is basically how the whole entire dog brain works. So ubiquitination regulates release at synapses. The first question that I'm going to answer is, what is protein ubiquitination? And then I'm going to talk about how it functions in the nervous system. So what is protein ubiquitination? This is the addition of a small protein called ubiquitin. It's 8 kilodaltons in size, and it's conjugated to the lysine residues in target substrates. And the addition of this small protein changes the trafficking, the activity, as well as the turnover of proteins. So that means that proteins actually get actively degraded into their amino acid components, and then they get rebuilt again. So this is important for all of these steps. There's all kinds of different signals. If you add one ubiquitin to a protein, that means something versus adding many ubiquitins or a polyubiquitin chain. And importantly, this is a system that functions throughout the body, throughout all of our cells, in a number of different pathways. Hence ubiquitin is ubiquitous, and has major roles in DNA repair, immune response, inflammation, and more relevant for neuroscience, it's been shown to have an important role in neurite outgrowth, in the remodeling of synapses, in response to plasticity or other remodeling changes. So protein ubiquitination is actually entire cycle. So it's a little bit more complicated than just adding the ubiquitin, and then protein goes downstream and it does some X activity. So it turns out that there are a number of enzymes involved in adding the ubiquitin to a target substrate, and these are aptly named E1, 2, and 3. So the E1 enzyme activates ubiquitin, E2 and E3 then together ligate the ubiquitin to a target substrate, and then substrate downstream is involved in some activity, or it gets degraded by the proteasome. However, prior to that, the deubiquitinating, or dove step, removes the ubiquitin in order for it to be degraded by the proteasome, or for transient modification or altering of the activity of the protein. And importantly, this is really not just the addition of the ubiquitin modification, but we know from diseases of enzymes in this pathway, et cetera, that this is actually needs to maintain an actively functioning cycle. So you have a certain amount of ubiquitin constantly being added and removed, and you need to maintain this free pool of ubiquitin. And each of these steps has important downstream consequences in the cell. So what I'm going to be talking about in the first part here, and actually throughout the talk, is pharmacological inhibitors of specific enzymes in this pathway. And the first important one is the E1 enzyme. So this is the first step in the pathway. So this is going to block everything from this point on. And there are two inhibitors that I'm going to talk about of E1, Xyram, and NSC, lots of numbers. And then the deubiquitinating enzyme inhibitors are G5. So there's also a lot of different deubiquitinating enzymes, but they all function through the same mechanism. So they use one inhibitor that essentially blocks all of them, and a more specific inhibitor, LDN, which specifically blocks just one deubiquitinating enzyme called UCHL1. So I just want to sort of add in this point to think about while I'm going through the talk here, is that, as I mentioned, this is a cycle, and adding or like removing the activity of an enzyme at one point in the pathway kind of stops the whole cycle from occurring, where it's not just like the removal or the addition of the ubiquitin, but it's really the dynamic change in the pathway. And I think that this has important implications for the effects of these drugs. And I also want to point out, before I get too further, that Xyram, an E1 inhibitor, is also a pesticide which is strongly linked to Parkinson's disease. So studies out of UCLA have found that thanks to California's very apt record keeping of chemical handling and health records, it turns out that agricultural workers that handle Xyram and various other pesticides, but especially Xyram, have an increased risk of Parkinson's disease, and particularly early onset Parkinson's disease. And just in case you're wondering, this is a pesticide that's still very actively used at a high rate. So don't worry, none of you guys have to start going organic all of a sudden. It's mainly a risk for the people that handle the drug, because this is when it's in its really highly active form, and it actually breaks down very rapidly in the environment. So by the time that we get our food, there's usually no traces of this particular drug or similar types. So what's interesting to point out as well is that in Parkinson's disease, we know that only about 5% of them are inherited. And so that means that 95% or so are of this unknown origin, or essentially of environmental and genetic background exposure. So, and one of the first sort of indications of this was actually the injection of, or several IV drug users had injected themselves with synthetic heroin analog that had an excess amount of a byproduct, which is turns out is MPTP, and they developed Parkinsonian symptoms within a week. So going back again, just to remind you of these drugs, we're going to talk about E1 inhibition and deubiquitinating enzyme inhibition and how these impact synaptic function. And the first thing I'm going to talk about is spontaneous activity. So how do we measure spontaneous activity? The little blue triangle guy here is my cartoon of a neuron. And essentially what I do is I have these cortical neuronal cultures from rats, and I patch plant them with small micropipette, and then I record the electrical activity inside the cell. And what I do is I add these drugs, tetrodotoxin and picrotoxin, which block the action potentials, or sort of like the evoked activity in the neurons and the inhibitory activity. And what that leaves behind are these small spontaneous events. And these are called miniature excitatory postsynaptic currents because they're little. And essentially, each one of these corresponds to just one of these vesicles fusing with the membrane and releasing its neurotransmitter content into the synaptic cleft and then being taken up by the postsynaptic receptors. So in a way, what we can learn from these kinds of recordings is whether or not there's a change in the presynaptic compartment or in the postsynaptic compartment. So what do I mean by that? Well, if we know that each one of these corresponds to a single vesicle fusing, then if we see a whole bunch more of them more frequently, then we know there's a lot more vesicles fusing, right? On the other hand, if we see that the size of them increases or decreases, that means something's happening to the conductance of the postsynaptic side. So this is a really good way to tell right off the bat, do we have something that affects the pre or the postsynaptic side? So what I do is I patch these cells, and then I add the inhibitors of action potentials and IPSPs, and I record the frequency of minis over time. And so these last for about 30 minutes, and you can see here in the control, this is relatively stable. There are some fluctuations. This is the frequency now of minis. And when I add Xyram, which starts at the black bar, we get a really rapid increase or enhancement of mini frequency. And this also happens in the presence of G5, the deubiquitinating enzyme inhibitor. On the other hand, if we look at the size of the amplitude, these change a little bit, but what is really striking here is that we see this very robust enhancement of mini frequency, which we think means that there's a change in presynaptic release. This also happens with the other two inhibitors that I mentioned. So NSC, again, is an E1 inhibitor, and LDN, which is a specific inhibitor of UCHL1 only, leads to an approximately twofold increase in mini frequency. So I showed you that inhibitors of different enzymes in the pathway seem to enhance spontaneous activity. Next thing that I want to look at is what happens to evoked activity and synaptic vesicle cycling. So in order to do this, I use a fluorescent probe called synaptofluorin. And essentially what this is the combination of a synaptic vesicle protein, so a protein that normally resides on the vesicle. In this case, it's going to be either V-glut, which is a glutamate transporter, or VAMP2, which I'll also talk a little bit more about later. And GFP, or green fluorescent protein, that has a shift in its pH sensitivity, such that inside the synaptic vesicle, where it's an acidic pH of 5.5, the fluorescence is completely quenched. But when you have stimulation or a signal to release synaptic vesicles at the axonal gluton, what happens is the inside of this vesicle becomes contiguous with the extracellular environment, which is a more alkaline pH of 7.4, and you get a strong increase in fluorescence. So basically what I'm going to explain, the experiments that I'm going to explain will show that, or will demonstrate changes in exocytosis or release of synaptic vesicle corresponding to an increase in fluorescence. And then once those vesicles get recycled back to inside of the cell, there's a reacidification or a decrease in fluorescence, and that corresponds to the endocytosis or recycling. So here's a video of what that looks like, and I'm stimulating with 20 hertz for 5 seconds. Should I turn off the lights? Sorry. We practiced this before, and I obviously might have screwed it up. So anyways, essentially what you're seeing is cortical neurons that are transfected with V-glut-1 fluorin, and they're being stimulated with approximately 100 action potentials. And when they get stimulated, all these vesicles fuse with membrane, and you see this great big lighting up of everything. Okay. So essentially what this looks like, and the different measurements that I'm going to be talking a little bit about, are the baseline fluorescence, and then you see this very rapid increase or change in fluorescence that corresponds to peak exocytosis, so that's the fusion of the vesicles. And then the decrease in fluorescence corresponds to the endocytosis, or the recycling of vesicles. And this corresponds to the stimulus. So what I do is I give this stimulation of 100 action potentials every 5 minutes. So 100 action potentials, rest 5 minutes, 100 action potentials, rest 5 minutes. I do this for 30 minutes, and at the very end, at the 30 minutes, I add ammonium chloride to the solution, which essentially collapses the pH gradient across all of the synaptic vesicles. And what that does is it reveals everything in that region, so all of the synaptic vesicle pool, essentially. And basically what that looks like is after the peak and the endocytosis, then we add ammonium chloride, and we get this very large increase that corresponds to everything that's present, all of the protein that's there. So this is just another sort of brief demonstration of how this experimental setup looks. This is a control condition where I am showing the baseline prior to stimulation, so there's a small amount of expression at the surface here, and then during the stimulus, there's a large increase in fluorescence. The arrows indicate the axonal boutons that I was talking to you about, those presynaptic sites. So they're sort of like these round little guys. And then if you look from the 5-minute time point to the 25-minute time point, there's actually a small or subtle increase in the baseline fluorescence, which you may or may not be able to tell from here. And this is important to remember as well. So there's a small amount of protein that's not totally retrieved from the surface. But we still see that there's an increase in fluorescence corresponding to stimulation. So essentially, just remember that we do see that not all the protein is retrieved, but we still see a change in fluorescence in response to stimulation. So what happens when I add inhibitors? Well, it turns out that E1 and deubiquitinating enzyme inhibitors reduce the synchronized or evoked release. And here we have a control trace where the black is the delta F over F, so that's the change in fluorescence over baseline fluorescence. So just to remind you, so the baseline is right before a stimulus, and then there's a change to the peak. And so this is relative to the baseline as well as the change. And then 5 minutes later, at that 5-minute mark, the drug is then added, and then 5 minutes later, we look at the response to the same stimulus. And in control, it's very similar. In Xyrem NG5, however, you can see the red trace, the second one, is already declined in size, and in NSC, by 5 minutes, the response is already totally ablated. So this is also shown in this plot here, where the black trace is the control, and this is relatively stable from 5 to 30 minutes, whereas in the different drugs, I apologize, I didn't put the color code on this slide, but this is NSC, Xyrem, and G5, and the evoked responses, or the delta F over F, decreases over time. But just to remind you, this is dependent on two values, the baseline fluorescence and the change from baseline. So what happens to the baseline fluorescence? Well, if you remember earlier, I said in the control condition, this actually increases gradually over time. So again, that's the black trace. But if you look at the experiments with the different drugs, we also see that these values increase over time, and more so, or they tend to increase more so than in the control condition. So what does that mean? Essentially, if the baseline increases and then you change in fluorescence, the overall value is going to decrease, right? So we think that there's a decrease in the response to stimulation, but we don't know if that's because there's less to be released or if that's because there's actually a problem with releasing those vesicles. So what I look at in order to differentiate those two possibilities is the F max, or the total fluorescence during stimulation. So this should incorporate the baseline and the change in fluorescence. So essentially, instead of saying how much does it change from baseline, just says what's the total amount when I stimulate, because everything that's on the surface is already fluorescent. And if you look at this value in the control condition, it actually slowly, gradually increases. If you remember, there's a little bit on the surface, and then there's the same amount that's being released over time. And if you look at either Xyrem or NSC, there's a significant decrease, at least in early time points, in this total value. So what that means is, at these times, there's a deficit in releasing synaptic vesicles. There's still something there, and it can't be released. And then later on, these values start to drift back up, which could be indicative of there being now protein stuck on the membrane. So the next thing I wanted to look at is whether or not there's a change in the endocytosis, or the recycling back from the membrane. And one way to do that is to look at the rate of the change from, so the rate of change from the peak to here. So you can see here in Xyrem, there's clearly a slowing of this rate. And in G5, there doesn't seem to be a measurable change. And with NSC, unfortunately, there's no response here, so we couldn't really tell this time. But what we can say is that there is a significant slowing of endocytosis, at least in Xyrem. So not only is there increase in fluorescence on the plasma membrane, but this is indicative of this protein actually being stuck there and being unable to be recycled. Then the last thing that I look at is, again, that ammonium chloride revealed pool, which is the change in fluorescence once you add this, the ammonia that collapses the pH gradient. And what you see is that compared to the control conditions, each of these drugs really drastically reduces that. So there's really very little left at the end. And this could be related to protein being stuck on the membrane, or it could be related to vesicles diffusing out from there. And so in order to look or to distinguish between these two possibilities, again, we look at the total fluorescence, which incorporates the baseline and the pool that's revealed. And when we do that, the Fmax shows that there's really no change between the conditions. So that means now we have a shifting of the protein from inside the presynaptic site to the plasma membrane, where it's either being stuck there, or as I showed before with the spontaneous release, there's so much release happening and it's just not being efficiently recycled back in time. So that's really just a summary here. So we see that there's an increase in mini frequency. So just to remind you, that means that more of these vesicles are spontaneously fusing with the membrane. There's a decrease in the evoked amplitude. That means when the neuron sends a signal that says, hey, it's time for you to release, they're unable to do that. Endocytosis seems to be inhibited, at least in Xyrem. So they're unable to go from here back through this cycle. And I didn't show it, but I have a slide at the end if anybody's really interested. And this doesn't seem to be due to a decrease in calcium. So the last thing that I'm going to talk about in the ubiquitination of synaptic proteins is one of the targets that we think might be involved in the change in release at presynaptic sites. And this actually came out of a proteomics screen that our lab did, postdoc Anna Caputo did with, in collaboration with the Wohlschlegel lab, where they took synaptic proteins, or this sort of preparation where they can isolate them, and then they treat them with either the E1 or the deubiquitinating enzyme inhibitor, and then send them to mass spec, and they look specifically at the levels of ubiquitination in the proteins, and then can compare to the non-treated conditions. Say, does the level of ubiquitin change, and which proteins? So one of the targets that came out of this is VAMP2, which is a snare protein that is critically involved in fusion events in cells. So it seems like a pretty good target. And of the snare proteins, there's VAMP2, which is a V snare or vesicle snare, and then syntaxin and SNAP25 are plasma membrane snares. So this, imagine there's this like circular synaptic vesicle here, and then this is a plasma membrane. And essentially these proteins like wind up really, really tightly, and there's a lot of other things involved, but when the conditions are right, they pull the two membranes in very, very close together, and it becomes energetically favorable just for the membranes to fuse together, and that's how essentially the vesicles release their contents. So we thought that this was an interesting target to follow up on, and we had two lysine residues that were identified in this screen, lysine 52 and lysine 59, and they tend to have some conservation across other VAMP isoforms, and also they tend to be pretty highly conserved among different species. So this is not just some random sequence as well. It turns out that this region of the protein is really important for snare fusion as well as endocytosis and synaptic vesicle targeting. The VAMP2 protein is comprised of 3 domains. The first N-terminal, or that's the amino terminal, is a proline-rich area, and then the C-terminal is the transmembrane, or that's the region that sits in the synaptic vesicle. And then between those 2 regions are these predicted alpha helical regions. And the alpha helical regions are thought to be really important in the coil-coil structure that snare proteins form with one another. This is a very tightly opposed, structure that keeps them very closely together. And then in the helix 1, there's predicted peptides, or there's predicted residues that are involved in fusion of membranes. And so these 2 regions, sorry, I got a little ahead of myself. So the first region is from approximately amino acid residue 30 to 50, and then the second one is approximately 60 to 90. And our residues of interest are at 52 and 59. So we think this is a really interesting juncture. So in order to look at the specific effects of these particular mutations, what I did is made use of that fluorine tool that I mentioned earlier. So the first one that I was talking about was based on VGLUT, which is a protein in synaptic vesicles for basically pulling neurotransmitter into the vesicles. And what we do now is use VAMP2, that fusion protein, and then have the pH-sensitive GFP attached to that. So we make point mutations so that those lysine residues are now arginines and they can no longer be ubiquitinated. And then we check to see, do they express in neurons, do they undergo exocytosis and endocytosis, and then what happens to the synaptic vesicle pool size and distribution. So the other thing about this is that there's slightly different experimental setup that I'll explain. And instead of doing 100 action potentials at 20 hertz, now I'm doing a larger simulation of 600 action potentials. And essentially what this means is that it's going to release a larger pool size, or the recycling pool at these synapses. So I do this 3 times now instead of over 30 minutes. And we'll look at the baseline versus the peak, as well as the peak versus the total synaptic vesicle pool size. So this gives us information about the amount of vesicles that are being released relative to the total that's present. So when I transfect neurons with these, they express, and I should have mentioned that's here. So this is the double mutant. I also made single mutants as well as the double mutants, but the single mutants turn out to be sort of in the spectrum of wild type to double mutant, and so what I'm just going to show you is the double mutant because that's the most interesting. And here's the regular VAMP2 expression. And I should also mention that this protein is more often localized to the surface, and that's why you see more fluorescence. So with VGLUT, it's almost all in vesicles, so there's very little baseline. Here, there's a lot on the membrane, so it's already fluorescent. So when we stimulate these guys, we see that they actually have a little bit more release than the wild type. So the red is the double mutant here, and the black is the wild type, and these are the different times, so 5, 10, or 0, 5, 10 minutes essentially. And if you normalize this value, they actually follow a very similar pattern. So again, there's a small amount of protein that's not being totally retrieved from the membrane. So the vesicles fused, there's still some protein, and then the next round doesn't change as much relative to baseline. If you look at the total pool size at the end, there's actually a larger pool in the mutant, which is potentially interesting. And if you compare this larger amount of exocytosis to the pool size, it turns out that they have the exact same recycling pool. So that means that even though there may be more vesicles in the mutant, they're actually still releasing the same amount, relatively speaking. So what happens with this larger pool size? Well, we thought this was potentially interesting and wondered if this means that there's a change in the localization of the protein. So we looked at the surface expression, and that being like the baseline fluorescence relative to that total pool size. And it turns out in the double mutant that this is decreased relative to the wild type. And so essentially that means that there's now more protein inside relative to outside. And these, each of these points are the mean of a total experiment. But if you look at and count every single bouton within each and every experiment and look at the ratio of surface to pool, it turns out this is a really significant decrease in that ratio. So what's happening now is VAMP2 is being redistributed into synaptic vesicles rather than at the plasma membrane. So conclusions and next steps. We think that VAMP2 may be involved, or the ubiquitination of VAMP2 may be involved in its distribution between the plasma membrane and the synaptic vesicle, and this may be related somehow to vesicle recycling or endocytosis. But does this relate to the findings that I showed you earlier? So with the pharmacological inhibitors, what we see is that there's a tendency to have the protein stuck on the membrane and not in the synaptic vesicles. So it's a little bit different, but we're not entirely sure at what state the proteins are when they're being stuck. So we know that VAMP2 cannot be ubiquitinated, but we also know that when we disrupt any point in the cycle that we seem to have the exact same effects. So it's not entirely clear if this is if the proteins, the synaptic proteins that are involved in release are ubiquitinated or polyubiquitinated or not ubiquitinated. So this is another thing that still remains to be determined. And then based on these findings, we think that there is an interesting effect here, but we want to know, will we still see this effect, or will we see a more significant or striking effect when we get rid of all the normal proteins? So we're adding this mutation to the wild type or endogenous background. What if we get rid of that endogenous protein and then we introduce the mutation? What happens? And so this is something that I've also been following up on using CRISPR to knock out VAMP2. And just as very early starting off proof of principle, it seems that the CRISPR knocks down the protein. I have no idea what the mutant does in this background, but these are just some experiments that I did stimulating neurons that have CRISPR against VAMP2 and then V-glutafluorine. So it's not VAMP, this is V-glutafluorine. And in the normal just V-glutafluorine background, stimulate with 100x potentials and you get this response. If I stimulate the CRISPR knockdown with 600x potentials, I get this response. And then the 100, which is similar to here, we get this, which is very similar to published reports where knocking out VAMP2 leads to approximately 1% of total evoked release. So we think that this is not just important indication of what synaptic protein ubiquitination may be doing, but using this technique to explore the ubiquitination of other synaptic proteins. So while we see potentially the VAMP2 effect may mimic some of, or may have something to do with the pharmacological phenotype, we still see that there's an enhancement of spontaneous release, for instance, and we don't see that with knocking out VAMP2. So very likely there's multiple synaptic targets. So what we would want to do ultimately is go through some of these other proteomics hits, which include the other snare proteins, SNAP25 and syntaxin, and also V-glut, and use these similar tools in order to study what what ubiquitination does in modulating synaptic release in this context. So I'm going to kind of shift directions a little bit, but not really, and talk about how pesticides modulate synaptic function, and this is a little bit shorter, so don't worry, I'll get you out of here in a reasonable amount of time. If you remember, I said earlier, Xyram is a pesticide which is linked to Parkinson's disease. So this is sort of how I got interested in how pesticides might affect synaptic transmission. We use it as an E1 inhibitor, but we also know that very likely it has other targets. In fact, there are a few others that are published, including aldehyde dehydrogenase and sodium calcium exchanger. And the other thing that I just want to bring up about Parkinson's disease is that most of the time we think about it in the context of the substantia nigra, which are dopaminergic neurons and are very important in fine movement control. And in fact, Parkinson's disease is a disorder of central nervous system as well as the peripheral nervous system. And patients or individuals that have Parkinson's almost always have other autonomic or sympathetic nervous system dysfunction. And these include hypertension, constipation, problems with thermoregulation, as well as olfaction. And there are these pathological inclusions called Lewy bodies, which are really characteristic to the dopaminergic neurons that die in Parkinson's disease. And these have been found in a number of different but very specific neurons throughout the nervous system, including adrenergic, GABAergic, cholinergic, and glutamatergic neurons that are important for these particular functions. So we wanted to know was, since we know that Xyrem has these very striking effects in our neurons, is what does this drug do to neurons? And can we look at it in another context besides, since we know that it affects glutamatergic neurons in our cortical cultures, what is it doing to other types of neurons, and can we see a difference between specific types? So these experiments were done that I'm going to first talk about in our cortical cultures, and this was done by a rotation student of ours, Caitlin. Did a great job of really thoroughly characterizing these effects, where we essentially patch these neurons, we inject amount of current, so there's no inhibitors now. This is not this spontaneous release, but we're actually inducing action potentials and then looking at those properties of action potentials to say, is there a difference when we add Xyrem and when we don't? And so then she records for a time, and then we add the drug and we see what happens to the properties of these action potentials. And then this little red arrow here just points to these events that tend to happen in Xyrem, where we have these inhibitory events followed by an additional action potential. So I'm going to just generalize or go through quickly rather than show you like a bunch of little dots that show that the membrane depolarizes. So that's this potential here when you're recording right before adding the current. So that tends to be more depolarized or more excitable. The envelope potential also increases, so that's the change once you inject the current. There's a smaller after hyperpolarization, so that's, when you have the action potential, and then it sort of resets back down. There's this refractory period or after hyperpolarization, and that actually decreases in size. And there's actually the most significant effect is there's a reduced time to peak. So all of these things are really indicative of an enhancement of excitability. And in some of the neurons that were recorded from, you see that it's really striking in that you just can't record from them anymore, or you lose all the action potentials, and some of them you just see these subtle enhancements over time. So essentially what the conclusion from these experiments is that we see a, generally speaking, enhancement of excitability in neurons with Xyrem. So we wanted to know whether other pesticides would have similar effects, especially because we know that there's this relationship between exposure and Parkinson's disease. And just to save a lot of time for all of us, what I found is that of the pesticides that we tested, the dithiocarbamates tend to have very similar physiological effects as Xyrem. So Xyrem is of this dithiocarbamate chemical class. And these are organosulfur compounds, and they're also used in rubber vulcanization. They have these disulfide groups that make them react with thiols. They're also metal chelators, they're very highly reactive, and they're lipophilic, which means they can cross membranes. It turns out that these dithiocarbamates tend to have common targets, including aldehyde dehydrogenase, and I'll show you that I think they also have E1 as a common target, and it seems that they have similar physiological effects. So when I showed you that increase in mini frequency earlier, what I did is I went back and looked at whether or not other pesticides or similar chemicals would do the same thing. And what I found is that other dithiocarbamates, including disulfiram and manab, lead to this really strong enhancement of mini frequency. So in this first one, this is actually 2 different concentrations of disulfiram. And this is interesting because this is a drug also given to alcoholics. So it inhibits aldehyde dehydrogenase, and this used to be given to people to sort of decrease their desire to drink because it makes them really sick. And happens to be very similar in chemical structure to Xyram. And there are also published incidents of people developing Parkinson's like symptoms after large exposure or taking a lot of this drug for a short period of time. Manab is another commonly used pesticide and also leads to this increase in mini frequency and has the same disulfide properties and also presence of metal here, which is manganese, and Xyram here, which disulfiram does not have. So there are some subtle differences, but we think that the dithiocarbamates seem to have this very similar chemical reactivity. They seem to have similar targets. So they've been published to all inhibit aldehyde dehydrogenase. And in the context of the experiments that I showed you earlier, the Xyram is an E1 inhibitor, so we wanted to see if they would also inhibit E1 in the same way. So this is a Western blot for E1 enzyme. That's the first enzyme in the ubiquitin pathway. And essentially what this is the enzyme by itself, and then the enzyme conjugated to ubiquitin, so part of that first step in the pathway. And when we add Xyram, it almost totally abolishes that E1 conjugated to ubiquitin. And you think that this is through a thiol-ester bond between E1 and ubiquitin. So again, these are proteins that react very strongly with thiols. And then when we add disulfiram, here's the lower dose and the higher dose, and then manab. What's kind of interesting is that we don't actually see that the second band is abolished, but it seems to be more stabilized, and then there's a third band. And we think that this is actually a stabilization of a state of E1 where it has 2 ubiquitins conjugated to it. So in the pathway from E1 to E2, there's a adenylated ubiquitin that then becomes a thiol ubiquitin, and when that is there, we get another adenylated ubiquitin, and this one is immediately transferred to E2. But these dithiocarbamates have also been described to dimerize proteins or create stable thiol bonds. And so my prediction essentially is that that's what's happening here. So we know that the dithiocarbamates, while they're not having the exact same effect, it's very likely that they have very overlapping targets. And so, and potentially similar but different physiological outcomes because of that. And so then this is just another example of the stronger band of, or sort of additional image of the third band in manab. And again, these are thiol bonds, so in reducing conditions, they're completely abolished. Then the last thing that I just want to talk about is dithiocarbamates affecting different neuron types. So most of what I've showed you thus far has been in glutamatergic neurons, and we wanted to know if there be a difference in an aminergic neuron type that may be more relevant to Parkinson's disease like system. And so in order to do this, we actually use Drosophila, which is the fruit fly, as a model organism because it has this really convenient neuromuscular junction that has both those glutamatergic neurons and actually aminergic neurons. So what I use for these experiments is actually in the life cycle of a fly, this larval stage here, and it has basically all it does all the time is like crawl through the food. So it's just like kind of contracting and relaxing, going through the food. And so if you examine the body walls, so this is a fillet prep where you like open it up and you kind of fillet it out and you take out all of the insides. Then those body walls, muscles that are involved in that movement actually provide a really unique tool for studying either glutamatergic or aminergic, which in this case is octopamine changes in response to drugs. And then these are some of the actual neurons that are sitting on top of the muscle here. So type 1, and they have morphological differences that you can identify them by. And another thing that's really great about Drosophila is they have a myriad of different genetic tools, so it's really easy to label specific things with whatever you want, and then just look for the changes in that type of neuron. So type 1 are glutamatergic, and type 2 are octopaminergic. And these octopaminergic neurons, it turns out, actually start spontaneously firing when they're exposed to Xyram. And this was initially discovered by a graduate student in David's lab, Sierra Martin. And here's a video of what that looks like. So these now are expressing GCAMP, which is the calcium indicator. So sort of like that fluorescent indicator I showed you before. He said these are responding to calcium, not indicating like fusion of vesicles. So you can kind of see that they're lighting up. And this is basically just washing on the drug and seeing that now all of a sudden there's spontaneous activity. And octopamine is the invertebrate equivalent to noradrenaline, and also an aminergic neuron type that is involved in learning plasticity as well as locomotion, this is the body wall, and egg laying. So we think that this is potentially interesting in the context of learning about how specific neurons are affected in Parkinson's disease. Now this may sound like it's getting a little convoluted. We're talking about locomotion and egg laying and fruit flies and things like that. But essentially what I think is happening is that Xyram and other drugs have these effects of enhancing excitability of neurons, and then based on the properties of those neurons, the intrinsic like physiology of them, the ion channel makeup, you're going to have a different outcome. So in this case, in these types of neurons, now all of a sudden we see this spontaneous firing. And then to this question, is this related to ubiquitin? We look at either dithiocarbamates or the other inhibitor of E1, NSC, and we see that this doesn't seem to be an effect of ubiquitination, but more so property of the properties of these chemical structures. And so this is disulfiram here. These are before adding drug. And this is Xyram. And here, this is 10 minutes after exposure to Xyram. And actually, this is 20 minutes after exposure to disulfiram. So there are some subtle differences. I think that probably has to do with the differences in chemical structure, but we think that very likely they have similar targets. So while the outcome is not exactly the same, the targets seem to overlap. So pesticides seem to, or these dithiocarbamates seem to enhance spontaneous activity. We showed you that at least with Xyram, that they alter evoked activity, endocytosis, and synaptic vesicle pool. They alter properties of excitability, and that this likely leads to different outcomes based on the neuronal cell type. This may have important implications for Parkinson's disease. So, sort of wrapping up from here, what I showed you today is that dynamic protein ubiquitination is important for regulating synaptic vesicle release. We think that this is a dynamic change in that there's removal and addition and active modification of proteins, and that this alters their activity very intimately in the fusion of vesicles and then the reuptake and recycling. And diethiocarbobates have similar molecular targets. They seem to be thioreactive, but as have been shown actually with other thioreactive chemicals is that they don't all act the same. So not every single protein that has a cysteine residue exposed is going to be affected or affected in the same way, but we think that there's a lot of overlap in targets. And so what I want to do from here is actually follow up on what those are. And based on those excitability experiments that I showed you earlier, it seems like potassium channels are a really strong target, and using the genetic tools of Drosophila, you can really easily take mutants of a specific channel and then identify where Xyram is acting on these neurons, and sort of identify from there what makes one set of neurons different from another set, and how does that give us information about the etiology and the course of neurodegeneration in Parkinson's disease. So with that, I'd like to make some acknowledgments. And the Schweitzer lab, we have a couple of undergrads that are working on the neuromuscular junction right now. I don't think they're here, but Logan and Janine done really great work. And people from the Schweitzer lab in the past who have helped me and contributed. Philip is an undergrad who helped me with making those mutants, the VAMP2 mutants. Yuki, who's here today, and she's been amazing help with everything in the lab, even though she's not even part of the lab, she still helps all the time. Anna, who's now a postdoc at UPenn, also did a lot of work on the Ubiquitin project and proteomics. I've had a tremendous amount of help and guidance from David Kranz's lab. Ciara, the graduate student who started the work on pesticides and Drosophila. Anna was also a graduate student and postdoc and really helped teach me a lot of basic fly stuff. Hiromi did some work on pesticides at the neuromuscular junction with me as well. And in Tom Odell's lab, Ryan did a bunch of experiments for me as well that I didn't get a chance to show today, but I have them later if anybody's super interested. And from Stephanie White's lab, Caitlin did some really awesome recordings with Xyram that I showed you earlier. And I just want to also take time to thank people in the neuroscience IDP. Jenny, who's a current SAO or student affairs officer, who's amazing, and past student affairs officers that I have to put up there just because they've all been just incredibly outstanding and really help facilitate things that otherwise would make graduate school so much more difficult. And then neurobiology, which is the department that I'm in, so yeah, it's neuroscience, neurobiology, I get it. It's sort of the same, but they're different, and these two people have really also helped make my life a lot easier, so many different occasions. I also want to take the time to thank my thesis committee, not member, but members. Dr. Felix Schweitzer, who's been my mentor for the last 5 years. I've learned a tremendous amount from, I've learned so much about basic physiology from, I learned how to patch from, and I think the first time that I patched a neuron, he told me the story of how you know that you're not supposed to mouth pipette TTX, but you'll be okay if you do. Just a little tingly on your tongue. So don't mouth pipette, but if it's TTX, you'll probably be okay. don't do too much. He's also been incredibly generous in like letting me do whatever I want in a way, and I not to say that like I've just gone totally nuts, but he's really given me a tremendous amount of freedom to figure out how to do things on my own and develop my own set of skills and my own techniques, and he's given me tremendous freedom to really like learn and interact with the neuroscience community. I went to the marine biological laboratory a few years back over the summer, and that's been one of the most amazing experiences in my academic research life. And if anybody ever has the opportunity to go there, you absolutely have to take it, without a doubt. I learned so much there, and I got to see so many amazing techniques and people and interactions, and it's also absolutely gorgeous. I really appreciate that, and going to conferences at SFN or Gordon conferences, and just that like encouragement to really go out and learn and figure things out has really just been sort of the real basis for who I am now as a scientist, and I really appreciate that. And also want to thank Dr. David Krantz, who's been an amazing mentor and really just like taught me everything that I know about flies and octopamine, and also thinking about like the entirety of science in the context of flies and octopamine, which is crazy, but like there's really a lot of applications you can take from there to learning or like being a scientist and understanding all of neurobiology from flies. You're really amazing. And I want to thank my other committee members, Tom Odell and Dr. Cameron Gunderson, who provided all kinds of amazing guidance and resources over the years for me, and really given me great feedback whenever I have committee meetings with them or whenever I have questions for them, and been really patient and just really, really nice to me for some reason. I really appreciate that. I also just want to like stop and acknowledge some of the amazing undergrads that I've had the opportunity to work with, because I've been so incredibly lucky. These are just a few of them, and I'd like to say that they've contributed to my projects, but in reality they're all like just incredibly smart and had their own independent projects, so they had nothing to do, well, not nothing to do with this work, but for the most part they kind of had their own things going on. Mitchell is actually going to be a grad student here in the fall in neuroscience, so yay. Philip, who actually did help me with the VAMP mutants early on, and then he went on and did some work with PC12 cells and capacitance measurements, which is just like something that I can't do at all. And Rudy, who is another summer student, who we worked on recording LTD in hippocampal slices and some of the effects of these drugs. And Janine and Logan, who I didn't have any pictures of, but they're also awesome and really smart. And DJ, who I've worked with a lot, and David's lab, and is just like the most insanely smart. He's like a graduate student already, and I think he's like only in his second year. So I just want to say that's been like a really enjoyable experience for me. I want to thank my family, all these weird, crazy people. My sister, Nicole, who's been really supportive and one of my best friends, and is the only family member that's streaming right now. And my awesome family who's here today, my dad and stepmom, and my really adorable nieces and nephews, and Christine, my stepsister, my mom here, and other stepsister, and other adorable nieces, and other furry relatives, my grandma here, and my nieces, and I'm just really happy to have their support and like happy that they haven't asked me to explain too much to them over the years. My extended family, my mom, my stepdad have been raising golden retrievers since me and my sister left home. So I have now this other like very extended relatives, and they're really crazy and cute. And I also have been like really fortunate to have some amazing friends in grad school, but also not the ability to take pictures of any of them. So I just have a few random pictures, but I have this really awesome neuroscience class that I've been a part of, and other neuroscience people that I had a lot of fun trips and adventures with, and friends that from working in David's lab and in Access, and lots of other people that I don't have pictures of, but are all super important, and I love you all. And then, of course, the most amazing and wonderful and loving and incredible, supportive fiance, Eric, who probably deserves this PhD more than I do. And I think I said it earlier today, but he could give this talk better than I can. So I apologize that you had to hear it from me and not from him, because he does a really good job. And he's got an amazing sense of humor. He's just like incredibly smart. He's been an amazing role model. He was also a graduate student here. He finished a year ago, and I'm really lucky that he's a year ahead of me, because basically everything I've done is try to like model what he did the year before me. So here I am, and I want to say thank you so much. And then you might have gotten a hint of some like furry other friends in my life, and so I just have to include my puppy because she's so adorable and she loves to help me study and help me cook, and we go on hikes together, and then she like does weird things to distract me while I'm working, but she's really sweet and cute, and so that's it.
Thank you very much, and I hope you've gotten an idea of how widespread the work of Kathy has been over the years. She's really sort of chosen her preparation without fear and sort of said, hey, this is an additional interesting thing that I'm going to explore, and she did, and learned enormous amounts of techniques, and got really awesome results, and I'm very proud and happy of you.
Thank you.
Any questions from the general audience? The family might have some questions, which usually isn't the case in this environment, but I sort of at least want to give you the opportunity. Anybody? Yes?
So ubiquitination is important for all kinds of other cellular processes. Have you looked at any of those and how they might play into what you see on the screen?
Yeah, that's a great point. And it's one of the advantages of using pharmacology is that I'm looking at very rapid effects. So it's a lot easier to compartmentalize those effects when we think about it on a very short time frame. And so the short answer is no. And the long answer is that there are several other studies that have looked at long-term effects of protein ubiquitin inhibitors and found that there are changes in plasticity. A lot of them lead to apoptosis or cell death over time. And actually, a lot of these are in like clinical trials or research for cancer therapeutics because they kill cancer cells. And ubiquitination is also implicated in cancer. So there have been studies that have looked at long-term effects, and the main effect is that we see cell death. And actually with Xyrem, there seems to be preferential death of dopaminergic neurons. So there may be some particular sensitivity in specific neuron types. But for my experiments, in a way, I kind of look at what I think is most likely to be acting in the time frame and in the compartment in the course of the experiment. So.
For your experiments with the point mutations in VAMP2, I'm wondering why, or if you did those experiments also, you're overexpressing those forms of VAMP2, but instead of conjugating the fluorine to that, conjugating it to the glutes initially to see how it affects that signal and if there's any differences.
So you mean like having V-glute and VAMP together?
Well, no, sorry. I thought you said in those experiments you conjugated the fluorine to the VAMP2 isoforms itself, right?
So it's like VAMP2 with mutation and then GFP. So the fluorine is actually GFP, which is already pH sensitive, but it's been shifted in its sensitivity. So that's what makes it the fluorine, is that it's more pH sensitive.
But with the initial experiments, you're using G-glute.
Yes.
GFP.
Right. So you mean if I did both at the same time?
Yeah.
So, yeah, that's an interesting question, and that is something that I thought about doing, is to say, like, is there a way that we could measure differences in like how VAMP2 mutants are acting versus some sort of more endogenous like protein that's targeted to synaptic vesicles, and that was one potential option, but I think because there's already sort of the like factor of the endogenous protein being there, that we thought that maybe what the next step should be is getting rid of that and then seeing what happens to VAMP2, because wherever like the VAMP2 mutant is going now, there's also probably going to be other normal VAMP2 protein on there, and so kind of looking at more of the extreme or like just total knockout background rather than adding another probe into the mix. But that is actually a really good idea, and something that I think would be an interesting way of teasing apart like two different pools of vesicles potentially.
More questions? Otherwise, again, you're all welcome in the botanical garden. If there are just like 2 minutes to cart everything down there and magically set it up. But it's a beautiful garden that we have for those who don't know it.
If I just have a question generically,
Yeah.
if for an entomologist standpoint, I mean, is it known, or do they sort of the mechanism by which these things kill pests?
So actually these so Xyram is generally considered to be a fungicide.
Okay.
And it's not known how it kills them. Yeah.
Okay.
But it could be in like some membrane trafficking pathway. But there are some other pesticides that the specific targets are known, but of the diethylchromates.
And certainly a lot of insecticides do target the nervous
Exactly.
system primarily,
Yeah.
so I'm not surprised in
That they would have effects on the nervous system of other species. Yeah. I think that's also a really valid point. And DDT and other organochlorides in a similar manner, they actually enhance, they decrease deactivation of sodium channels. I think they inhibit GABA receptors. There are others that are also insecticides that are anti-cholinergic. So there's a lot of similar targets or overlap there.
More questions? Or afterwards, then in the garden.
Yeah.
Thank you.
my calls with you. Okay. And I'll see you over there? Okay.
Thank you.
The long not seen one. Hi.
I get crazy busy.
But I hear you're going to Puerto Rico.
Yeah. Isn't that crazy?
No, that's wonderful. You're going to
it's not
Sunday.
Just
No, that's
gone for like
wonderful.
2 weeks and a couple of days.
All right.
Yeah, I'll go help you.